Thank you, Dr. Green. Today, I'll be presenting my graduate project, which is focused on defining non-coding regulations of von Willebrand factor expression using engineered endothelial cell models. The broad question is whether intronic regions within the VWF locus can influence von Willebrand factor expression. I'll start with some background on VWF biology and why VWF levels matter clinically. And then I'll move into the rationale for looking at why we looked at non-coding regulation specifically, and then describe the work done so far to establish a model and nominate some candidate regions. VWF is a critical hemostatic protein. It has three major functions that are most relevant here. First, it mediates platelet tethering by binding exposed subendothelial matrix and platelet GP1 beta alpha under shear stress. And this is especially important in high shear vascular environments where platelets need help attaching to the vessel wall. Second, VWF supports platelet-platelet interactions and aggregate growth through interactions with alpha 2b beta 3. Third, and also importantly, it protects factor VIII, a critical protein involved in the coagulation cascade, from degradation in circulation. Without VWF, factor VIII is significantly less stable, and plasma levels are heavily reduced, which contributes to the bleeding phenotype seen in more severe von Willebrand factor deficiencies. VWF is also structurally unusual. It is a very large protein with a mature sequence derived from a 2,813 amino acid precursor. It is a heavily glycosylated protein that dimerizes and then multimerizes, and those multimers are central to its hemostatic function. Larger multimers are generally more hemostatically active because they are better able to bind platelets and matrix under shear. This multimerization process is also linked to Weibel-Palade body formation in endothelial cells where von Willebrand factor is stored, which we'll see now. So VWF is synthesized as a precursor that includes both mature VWF and the VWF propeptide. These are produced in a one-to-one relationship and then processed during biosynthesis. VWF then dimerizes, multimerizes, and can be packaged into Weibel-Palade bodies. There are different routes of secretion, though. Constitutive secretion often occurs continuously and is generally associated with smaller multimers, while regulated secretion releases VWF from Weibel-Palade bodies and is associated with larger, more hemostatically active multimers. These larger multimers are also the main physiological target for ADAM-TS13 processing. Under shear, they unfold and expose their A2 domain, allowing ADAM-TS13 to cleave them into smaller, less adhesive forms, which is a critical component of VWF clearance. For my project, the important point is that the endothelial context matters here. 80% of VWF is synthesized within endothelial cells specifically, with the remaining amounts being synthesized in megakaryocytes. So its expression is connected to endothelial transcription, storage, and secretion biology, and thus a relevant endothelial model is needed. However, plasma VWF levels are not simply determined by one process, they are multifactorial. There is a transcriptional component, meaning how much VWF mRNA is produced. There is a storage component because VWF is packaged into Weibel-Palade bodies, and there is a secretion and clearance component as well. My project is focused primarily on the first of these transcriptional regulation factors and downstream cellular VWF output. Notably, when I measured this, I will be only taking one-time snapshots measurements of VWF levels, so I will not be modeling systematic clearance at all, which could play a large role in some phenotypes, but it's a limitation of our model. The reason VWF regulation is clinically important is that VWF level behaves like a hemostatic imbalance. At the low end, reduced VWF is associated with bleeding, including von Willebrand disease, and one of the methods of diagnosing this disease involves testing a patient's plasma VWF levels. And with a result below 50% of the normal standard, or 0.5 units per milliliter, indicating clinically low levels of VWF, which affects about 1 in every 1,000 people, and this is also called type 1 von Willebrand disease. At the high end, elevated VWF is also clinically important. VWF levels above 2 units per milliliter or 200% are associated with thrombotic risk, including arterial and venous thrombosis. Therefore, a careful balance must be struck, And too little VWF predisposes a patient to bleeding, while too much VWF contribute to thrombosis. And that is why the quantitative regulation of VWF expression is so important. So type 1 VWD is the most common subtype, and it accounts for roughly 80% of VWD cases. It is mainly a quantitative phenotype where VWF levels are reduced rather than the protein being completely absent or structurally abnormal in some other way. However, coding variation does not explain all quantitative VWF phenotypes. Around 35% of type 1 cases remain without a clear explanation from pathogenic coding variants. That creates a genetic gap. Some of that gap may reflect limitations in variant interpretation, complex inheritance, modifier loci, or other environmental factors, but it also raises the possibility that non-coding regulation could contribute to VWF level variation. The diagram on this slide is showing a Manhattan plot indicating the association of various loci with VWF levels. You may notice that VWF itself is not even the most strongly associated locus. As I mentioned, VWF is highly glycosylated, and people with an O blood type lack the A and B antigens that help prevent VWF proteolysis by ADAMTS13 that I mentioned previously. Hence, the extremely strong association with VWF levels. There are also other genes identified here that have an obvious causal effect, such as STXBP5, which is a regulator of VWF storage and secretion. Now, these signals tell us where variation may matter, but they do not directly tell us any causal relationships or what mechanisms are involved, except in some of the obvious cases that I mentioned, which is what projects like mine are intended to solve. One reason this could be plausible is that most of the genome is composed of non-coding sequences, which are relatively unexplored. Only around 2% of the genome itself is a protein coding sequence, and the other 98% includes introns, and as part of that, enhancers, promoters, insulators, untranslated regions, and other regulatory or structural elements. This matters for VWF because the gene itself is incredibly large. It contains 52 exons across 178 kilobases, and as you can see in the diagram, a massive portion of the gene is intronic. VWF is also a highly polymorphic gene, which introduces more opportunities for investigation into variants and other such things. My project is therefore focused on that region of interest, that intronic regions within VWF may contain certain enhancer-like regulatory elements that could influence endothelial VWF expression. However, I think it's important to briefly touch on how introns can specifically influence gene expression. As I'm sure we all know, introns are removed from mature mRNA through splicing, but before that, they are part of the transcribed sequence and can contain regulatory DNA elements. There are a few mechanisms here that I think are relevant. Intronic enhancer elements can bind transcription factors themselves, and through chromatin looping, these intronic elements can come into contact with the promoter or other regulatory regions, even if they are not immediately adjacent. In fact, chromatin looping allows even extremely distant elements to come into contact. In our lab, we have evidence of a superenhancer region over 40,000 base pairs away having a measurable influence on VWF expression. Chromatin accessibility is also important. DNA is packaged around nucleosomes, and regulatory proteins cannot bind efficiently if the relevant sequence is buried in closed chromatin. Open chromatin, however, reflects regions where nucleosome positioning, displacement, or remodeling makes the DNA more accessible. And if an intronic region is open and marked by regulatory chromatin features, transcription factors are more likely to bind there. Finally, introns are also part of the splicing context, so intronic sequences can affect expression through transcriptional regulation, RNA processing, or both. For this project, I am most focused on the possibility that enhancer-like intronic elements alter VWF transcriptional output in endothelial cells. Finally, getting into my aims. Aim 1 is to benchmark and validate 2 viable cell populations. Before getting into any genome editing, we need to know whether the primary endothelial populations are usable, whether VWF expression is stable enough to interpret across passage number, and whether there are any obvious pathogenic or confounding VWF variants in our cell populations of choice. For this purpose, we acquired two donor-derived primary HUVEC populations to work with. Aim 2 is to prioritize candidate regions. For a gene that is 178 kilobases long, this is a necessary step to filter through a wide range of potential areas that could influence and act as regulatory elements. This uses genome browser evidence primarily. Functional genome tracks and alpha genome predictions are also being used to select intronic regions that are most likely to affect VWF output downstream. And as part of this aim, I am also sequencing the candidate regions that we aim to test once they have been deemed as good enough targets for editing through our in silico analysis, and we'll be designing guide RNAs for later use in aim 3. Aim 3 itself is the downstream causal test. We are going to delete candidate regions using CRISPR-Cas9, and we're going to quantify the VWF transcript and protein output. Overall, again, my hypothesis is that candidate enhancer-like intronic elements within VWF will measurably modulate its expression. And then I'll start with aim 1. So why exactly are we using HUVEC specifically? First, VWF is primarily an endothelial product, as I mentioned, so using an endothelial model is very important. And venous endothelial cells specifically are generally more robust VWF producers than arterial endothelial models are. Second, these cells can provide endogenous readouts. We can measure VWF mRNA and protein without any manipulation to force them to produce it as it takes in some other types of cells. And third, primary cells are more physiologically relevant because immortalized or transformed cell lines can have some very odd behavior compared to what we actually see in the clinic. And that is very important for regulatory DNA specifically because enhancer activity and chromatin state are highly cell type dependent. The unfortunate trade-off is that primary cells are much harder to genetically engineer and especially recover after this genetic engineering, which I will discuss more in depth later. So as I suggested, our experimental system includes two primary HUVEC populations. We have a male and a female population, and they both underwent the same process of baseline characterization. The main reason we started with two populations is that we wanted redundancy against potential issues that could be encountered far downstream. It's possible that we could be months into an experimental pipeline only to discover that one of the populations carries some pathogenic variant in an intron or something that is of unknown significance, at which point you would really need a backup to not have wasted all that time. However, at the same time, carrying both populations through every downstream CRISPR experiment would double the workload. And so part of aim 1 is also to determine whether one population specifically should be prioritized if we reach a point at which we think that might be necessary. So characterization was necessary for four main reasons. First, we need an exact sequence in order to design guide RNA and for our genotyping and primer design. If there are any variants in the PAM sites or the guide targets that we're trying to target, then the experiment will not function at all. Second, we need to understand the passage stability, because if VWF output changes substantially across passage number, then downstream comparisons after editing will become much harder to interpret. And especially since if we're growing up some of these cell lines from only a few cells, then it may require several passages to actually get up to an amount of cells where we could do some analysis. So we need to know how passage number could affect it. Third, we also need a baseline phenotype. In order to spot obvious outliers in our results, we need a matched pre-edit reference, though obviously we also need to carry a control population through every experiment. Fourth, we need to screen the donors for the reason I alluded to earlier, that a single pathogenic variant or anything could be cause for instant exclusion. So our aim 1 workflow had four parts. First, the VWF exons were sequenced for pathogenic or confounding variants. We measured mRNA using qRT-PCR at passages 5 and 10. We measured protein with ELISA in both the media and the lysate at passages 5 and 10 as well. And we looked at ICC morphology as well using, I can't remember what that machine is called, but it's something. Getting into the results from our sequencing, I'm going to note here that the variants we're referring to are differences between our sequence cell populations and the reference genome assembly GRCH38. So the male line had 23 variants, including 14 intronic and 9 exonic, and the female line had 20 variants, 13 intronic, 7 exonic. There were 8 variants that were shared between the two populations, 6 intronic, 2 exonic. And importantly, there were absolutely no pathogenic variants of any kind discovered, except the one intronic BWS in the male line. But upon further investigation, it did not seem very likely to actually be pathogenic. Um, for whatever reason, it was still classified that way by Franklin. But I want to especially note that the result that we got from this was not very surprising because VWF is such a large and highly polymorphic gene. And with 52 different exons across a large genomic region, we would expect to find many different variants like we did. And I also want to point out that even though we only sequence the exons and through our primer design catch about 100 base pairs at most on either end, we still ended up finding more intronic variants than exonic variants, which kind of, suggests just how much variation there is in the introns within BWF. Then we're looking at the baseline QRT-PCR results that were used to evaluate the mRNA transcript stability across passage number. The main result here is that VWF mRNA appears relatively stable across passages 5 and 10 in both populations. There are visual differences, especially in passage 10 there between the male and the female, and there's obviously the strange happenings in that one well. This is an N of 1 on the male P5. But I think this was a result of human error, and I'm going to be repeating it soon. And otherwise, the results are quite tight between the experiments. And that is important because QRT-PCR is likely to be one of the cleaner downstream results after editing. And if an intronic deletion changes VWF transcription, then QRT-PCR should be able to detect that effect, provided the experiment is properly matched. So from the mRNA side, at least, both populations look usable. This slide shows how VWF protein was measured in conditioned media and then normalized to the total protein. Compared with the QRT-PCR, the results are obviously a lot more variable. There's a significant difference between the male and females at passage 10, and the females at passage 10 are also different from their passage 5s. And visually, there's a similar trend at P5 between the male and the female, with the male being higher as well, although it obviously was not significant. This doesn't entirely invalidate our female population, but does mean that the protein output is a lot more sensitive to donor and passage effects. And so for any downstream experiments we do, the ELISA comparisons will need to be matched very carefully and interpreted with caution. It is also an important piece of criteria against continuing with the female population specifically. And this is the protein lysate readout for VWF. There's also more variability than the QRT-PCR, but there are obviously no significant differences, and the pattern is not exactly the same as the media measurement. I also want to note the change in magnitude here. In the lysate, we observed around 3 to 4 times as much VWF as in the media, so suggesting that most of the VWF we're looking at remains cell-associated rather than secreted in our tissue culture conditions. For the downstream project, this means that media and lysate are complementary readouts. A regulatory perturbation might affect total VWF production, but the differences between the media and the lysate could reflect storage or secretion-related effects, so it might be hard to interpret just exactly how such a modification affects the output. However, the difference in magnitude between them means that we might be able to easier discern changes in VWF expression in the lysate specifically, just because the order of magnitude is larger. Moving on to cell morphology, this is just a simple picture of the P5 growths cultures at 40 times. They look like normal HUVEC cells. There are a little bit of qualitative differences. The females appear a bit more elongated, while the males are more circular and cobblestoney, but neither population looks abnormal beyond a certain margin that would make it unusable. So I also only included P5s here. The images are very similar in P10 as well. The same trend occurs with the qualitative differences. The ICEC as well kind of shows a different picture. The channels here are showing DACO anti-rabbit VWF staining in green. We're using red phalloidin for actin filaments, which are pretty hard to see in the male P5s, but they're a bit more visible. I don't know if the lights are making it hard to see, but the actin filaments are a lot more visible in the female cells. And the nuclei stained in blue here are very similar. So again, we do see another difference similar to the ELISA in the media. The males appear to be producing more VWF. They have a lot more green, but again, in the actual lysate, we didn't observe a significant difference here, so it could just be sampling bias or some other thing that went wrong, or an artifact. So we have three major conclusions to make from AIM-1. First, both our donor populations remain viable. Sequencing did not reveal pathogenic coding variants, and neither of them showed exceedingly low levels of VWF output. But the male line appears to be the leading candidate going forward, just due to the small differences in the VWF in the media, and because we may need to choose one at some point, and that's all we have to go off at the moment. And the third conclusion is that the protein output appears more passage dependent than the QRT-PCR and just in general was more variable than what we observed with the transcript. Overall, though, we have produced a usable starting platform for the endogenous VWF genome engineering studies. And on to AIM-2, we are looking in silico at our region candidates for editing. So AIM-2 has three major components. First, we start with our functional genomics analysis. This is just human inspection of the relevant genomic tracks with a focus on histone acetylation and DNase hypersensitivity to identify regions that look regulatory in endothelial contexts specifically. Second, we have our alpha genome prediction. This is using a machine learning model that I'll get into a lot more later, but here regions that we identified previously in the prior step as potentially regulatory are modeled as candidate deletions in silico, and then we can look at the predicted effects on the VWF RNA-seq signal and compare and contrast between that and the reference and score them. The third is the sequencing and guide RNA design. Once we have a candidate region that we've prioritized using the other two steps, the local sequence needs to be confirmed in the donor population, again, because we need it to design guide RNAs, and then we can design genotyping primers and such accurately. Thus, AIM-2's primary goal is to produce a promising candidate region for deletion and to set up and design the reagents necessary for AIM-3. And this is our starting point for candidate prioritization. The VWF locus can be viewed in a genome browser tracks, including the histone acetylation and DNase hypersensitivity tracks that I mentioned previously. Histone acetylation is commonly associated with active enhancers or promoters, while DNase hypersensitivity indicates chromatin accessibility. These tracks can all be viewed in the context of a specific type of cell only, in my case, HUVEX, although this specific image from our grant is not filtered to just HUVEX. They are the light blue and green. Sorry, I should be using the mouse. The light blue and green you can see here are the HUVEC specific ones, I believe, from the other images. So if a region is accessible and has enhancer-associated histone marks, it becomes a candidate regulatory region. I've put another blue square around this one specifically. This is intron 3, which is a particular region of interest, and this is the area of the project I'll be focusing on next. So I will want to point out here that these are just correlative studies. They tell us that a region has regulatory features in theory, but they do not prove that the region controls VDOF expression in any way. And without the use of future tools, it is difficult to narrow down these predictions from a larger, potentially regulatory entire network, say 10 kilobases, to a smaller fragment of interest, 1 kilobase or so, without guesswork or extensive wet lab work creating cloning these constructs and going through all these experiments. And that is where alpha genome comes in as a second layer of prioritization. I'll try not to go into too much depth here, but at a high level, it's a sequence to function machine learning model. It takes a megabase of DNA sequence as input, and it predicts the functional genomics outputs, including the tracks that we just saw in the genome browser, things like RNA-seq, DNase, hypersensitivity, etc. This model was trained on these in large functional genomics databases, primarily in code. And so it learns statistical relationships between DNA sequence and measured regulatory outputs across hundreds of different cell and tissue types. Some of these numbers of how many different contexts they use are shown in the bottom right of the image there. Um, and they also did this to a resolution of one base pair for a majority of the track types. Notably, for my purpose, it was trained on HuVex, and it can be run using a HuVex-specific accession number, though obviously it was not trained on our specific HuVex from our donors. A key advantage of AlphaGenome for our non-coding regulation is that it has been trained specifically to look at long genomic contexts. And that matters because, as I discussed earlier, some regulatory elements do not act on promoters and genes directly adjacent to them, and some of them can act on genes tens of thousands of kilobases away, and AlphaGenome can account for that. For this project, I will use AlphaGenome to compare a reference VWF locus, the GRCH38 that I mentioned before, against altered versions of our sequence from our candidate regulatory regions and see what happens when they are deleted. Here I'm just showing an SNP because it's easier to display, but the output that we get is the predicted assay score of the RNA-seq, DNase hypersensitivity, and histone acetylation, and the other tracks that we saw previously. So it's not important exactly what the absolute scores are here, but it does allow us to directly compare what it thinks the output would be from the reference and what would happen when we delete it. So we can get an idea of what these constructs would look like in a wet lab. Of course, this is in no way a causal tool, and it doesn't replace the wet lab work. We can't make any conclusions off of it, but it helps to save us some time potentially in not running many, many different constructs and maybe speeds it up. So this is an example of what I was talking about with Intron 3. This is a zoomed in version of 5 kilobases, a human prioritized subregion of Intron 3 that we looked at previously. There is a, I think, purple line there. I can't really see the color or remember, but that's where an SNP was identified, RS112814955, in a variety of GWAS studies, and it was shown to be heavily associated with VWF levels. And as you can see, this region, the VWF locus, has bountiful functional genomic evidence, lots of VWF, or lots of DNase hypersensitivity, and lots of histone acetylation. The second image on the bottom shows AlphaGenome's prediction of a 600 base pair deletion in that same region. After deleting the highlighted base pairs there, this box is showing it. There's a cutoff here for the histone acetylation because the resolution for AlphaGenome for that track is only 128 base pairs, so it's making an approximation. But as you can see, the DNase hypersensitivity is completely wiped out with the red reference still being there. Obviously, we deleted it, so the blue is gone in the highlighted region, but even outside of that, the red peaks are much more easily visible. And in the histone acetylation, you can tell even in the surrounding regions, it has basically been cut to zero. So the aim here was to refine a broad candidate region that was identified by human eyes into a more specific sequence that could realistically be tested by a CRISPR-mediated deletion. And therefore, the workflow is to identify a plausible regulatory region, define certain windows, and iteratively test them. We can run 50 of these at a time to see a variety of different potential results and fragments. And then we can ask which of these windows AlphaGenome predicts to have the highest effect on VWF RNA-seq output. I then used AlphaGenome to rank, as I said, a variety of deletions in this area. And the top predicted one was that 600 base pair region that I just showed you. It predicted about a 16% total reduction in the expression of VWF through the RNA-seq results from its machine learning. And this region, I should note, also contains the SNP identified in the previous GWAS studies, so it's also important for that reason. I will note it's not a massive predicted effect. 16% may be difficult to tell in some studies, but that's kind of expected for a regulatory element. That's why we're partly going with deletion instead of CRISPR-I, which I will touch on now. So I've already said I plan to use CRISPR, and one possible alternate strategy you can go with is using CRISPR interference instead, or CRISPR-I. It uses catalytically inactive Cas9 to repress local regulatory activity without cutting or removing the DNA sequence. So why am I choosing to go with active Cas9 and non-homologous end joining instead? The reason is that the predicted expression output may be modest, as we just saw, only 16%. If the true effect is that small, then a physical deletion of the candidate region would probably be more likely to show a statistically significant perturbation than an inactivation. And deletion also creates a direct genotype. We can ask whether cells in these um, deletions, if we want to go downstream and do future edits, or if we want to look at the SNP specifically, well, not in this case, but if you want to do a second deletion and compare haplotype style permutations, then you can do that, which you can't really do with CRISPR-I because it's transient. We will consider CRISPR-I as a possible future approach, but paired guide style deletion is the initial strategy that we'll be proceeding with. For our aim-through workflow, we have 3 or 4 major steps. UVEX will be co-transfected with paired guide RNAs, and the sequence confirmation and guide RNA design would be completed through aim-2 earlier. Second, flow sorting as an optional step for enrichment, depending on the observed transfection and editing efficiency. Since our plasmid that we of choice includes GFP, we can use GFP positive cells to sort for enrich for cells that have received the plasmid. Third, PCR screening will be used to estimate edit efficiency. The plan is to use PCR across the candidate deletion range and quantify band intensity with ImageJ as a rapid initial screen for the edit efficiency. Fourth, VWF will be quantified by QRT-PCR and ELISA in match conditions, as we saw in AIM-1 before. And the goal here is to link the genotype to the VWF transcript and protein output. This slide clarifies a bit more of our plasmid strategy. We'll be using the PX458 from the Zhang lab that includes the GFP, as we said. And our plan is to use 2 plasmids, each containing 1 guide RNA, and co-transfecting them at the same time. I'm revisiting the workflow a little bit here, even though we went over it, because I want to emphasize the main limitation of this project, as with many different CRISPR projects, is edit efficiency and recovery. Primary HUVECs are extremely difficult to genomically engineer, and they are especially difficult to recover as pure single-cell edited populations. It is likely impossible to recover them as a single cell. Our plan, therefore, is to nucleophect paired guides and optionally sort them, as I said, recover those surviving pools, screen them, and expand them. And the reason that flow sorting is only optional here is that nucleofection and low cell seeding in a 96 well plate are already very stressful events for HUVECs, and adding a third stressful event in flow sorting may simply make them unable to survive. But if our edit efficiency is too low to proceed with, obviously, we'll have to consider that, but it's optional for now until we see some early results from our edit efficiencies. This is all not as clean as just working in an immortalized cell line, but it's more physiologically relevant, and past experiences in the lab with immortalized cell have not shown good results for VWF output, so we will be trying to use primary cells. And then finally, linking the slide back to, or the study back to human genetic variation in VWD, as we saw at the start. These GWAS studies that we have give us a signal and show us where to look. We can use our in silico analysis to decide what to specifically perturb, and then do our wet lab experiments with our deletions to see the impact it actually has on VWF expression. It does not immediately solve any patient diagnoses, but it's the kind of mechanistic step that can help us move from simply having 35% of type 1 cases having an unknown cause to maybe being able to assign some of them to non-regulatory elements instead and variants there. So there are three main takeaways from our project so far. We have prepared our viable endothelial platform. We have our two donor-derived primary HUVEC populations that have been sequenced and benchmarked, and both seem usable at this time, though the male cell population appears to be the leading candidate for now. Our in silico analysis suggests a possible route for getting results for a confusing clinical genotype. A substantial fraction of those quantitative phenotypes, as I said, remain unexplained. And the evidence that we've seen, for example, the 16% predicted VDF expression suggests that introns could be possible contributing factors here. And lastly, intron 3 is the first region that I'll be analyzing moving forward. Functional genomic evidence and the alpha genome predictions show that this specific subsection of this intron are promising, and we'll be using paired guide deletion to test that in the future. Ideally, this project will move us from predicted regulatory DNA elements toward direct functional testing and be able to explain some of these variants. I'll end by thanking my supervisory committee, Dr. David Lillicrap, Dr. Cynthia Pruss, and Dr. Lois Mulligan. I'd also like to thank some of the members of the Lillicrap Lab, especially Natalie and Colleen, for their support and contributions to the project. Thank you, and I'm happy to take any questions.
Questions for Connor? Marco, go ahead.
Great work, Connor. I had a question about, I guess, your targeting strategy for intron 3. Is the selection of like deletions in intron 3 informed by intron 3 deletions actually happening in the population, or is it more so you notice that there's these trends in, like, I guess how there's sequencing predictions and responses in that region, and then you're knocking out intron or deleting parts of intron 3 to contextualize any other kind of like a single nucleotide polymorphisms in that region?
So Marco asked if we were looking at large scale deletions because they're seen in the populace, or if we're just trying to cover kind of a range of SNPs. And I don't think there are studies showing that there are common large scale deletions of introns. It's more that, I mean, we're already looking at kind of margin of error level differences in VDOF expression. So going straight to an SNP style deletion probably wouldn't show a big difference. It might, but it seems safer to start with a larger deletion in that case.
So if I can just follow up on that. So that's a SNP, an intron 3 that you're looking at, but it's associated with in the GWAS with what degree of difference in VWF expression, and what is that at the protein or RNA level?
I think it was like a p to the minus 20 or something VUF level, but it's possible that it alone could show a difference. But
so when you look at this with alpha, whichever program you're using, it's not a splice junction, presumably.
No.
It's well into an intron. And I'm assuming then that there are no recognizable known data binding cystathionine elements that it is within.
DNA binding elements like transcription factors?
Well, no. Well, just known sequences that DNA binding proteins are known to interact with. Transcription factor bindings.
I think there are some in the area. I don't think none of them overlap with the specific 600 base pair region that we were looking at, but there are some in the region. Yes. Not on the SNP itself, no, but there are some in the region for transcription factor binding sites. I can't remember the DNA binding site specifically.
And do you know if anybody has tried mobility shift type assays to see evidence that there's anything that binds to that sequence?
Not that I'm aware of, but
that's an assay that your lab is very familiar with from years gone by. Yeah. Anyway, other questions? Go ahead.
Really nice talk.
Thank you.
I was wondering on slide 16, and you're talking about like the different variants there. How did you know that none of them were pathogenic? I don't know if I missed that there.
Sorry, most of them, Tynan has asked how we knew if the variants were pathogenic or not on the sequencing overview. Most of them are in Nomad, just they're common enough, like 90% in the population or something, that we know they're benign. There were, I think, 4 in the mail that I had to check with Franklin, of which 3 ended up being benign anyway, except for that one intronic BWS that they were unsure about.
Sorry, just
Marco?
follow up on this. I mean, Nomad is nice, but then like it can suggest things are benign because they're
Yeah.
very common. But have you cross-referenced that with like a QTL data set to actually compare the effects of these SNPs on BWF expression specifically?
Marco asked if we've cross-compared with QTL
It's
expressions.
like a GWAS
Yeah.
linked with
Okay.
gene expression. So like it takes like a GWAS variant and instead of associated with like VWD or something, you could directly correlate those SNPs with VWF expression, which I guess would be kind of your phenotype of interest.
I can't say we've looked at that yet, but that's an interesting avenue of exploration.
You know, it's
Quantitative trait loci.
that's it.
It's like, but it's just instead of the like disease trait, you're using like expression of, you could go genome wide or transcriptome wide expression.
Any rational.
VWF. Yeah.
Maybe you can tell us a little bit more about the CRISPR-I, because I think there's lots of different ways that people do that, and what's the tool that you're planning on using there? Is it something that brings down a suppressor, a transcriptional suppressor?
We did not look greatly in depth into CRISPR-I for the reasons that I gave. I think we were looking at CRAB as a potential option, but I can't say that we looked in depth at ways to conduct CRISPR-I. We figured that the deletion would be necessary in order to see differences.
Because I'm not sure, maybe it's a good idea that you're not looking, because I'm
Oh.
not sure if that's going to be a fair way. If you're pulling down a repressor, a transcriptional repressor, then you're kind of stacking the odds against it. So in theory, you probably could target any particular region. So whether that's going to inform that particular region of the genome as being a place where transcriptional regulation is going on in the group. Other questions? Chris first?
So Connor, it's a great talk, but forgive me because I'm not an expert in the field. So if the goal is to try and figure out enhancer elements and I get in the intronic regions, not you know, 5' or 3'. My limited knowledge of people who have done similar type experiments have taken sort of their gene of interest. You've got 52, and that's a large gene, chunked it up and put it onto some sort of order to try and figure out if they had or didn't have that specific region, they would see expression go up or down or not go up. But you're working the other way to try and like minimally sort of reduce, and I guess this is kind of what Peter's comment was. Is there a way to kind of maybe complement what you're doing to try to, I guess I'm lost on why that particular region around 3 was validated as something to go after first. That was a long question to say. Can you just remind me again why you chose that region in 3 as an area of focus to work on? And how that's, and I guess how that's going to help you identify the enhancer regions that you're trying to find.
Okay, Dr. Nicol has asked why we picked intron 3 specifically. The very simple reason was just that like these 3 red boxed areas are very clearly have a lot more histone acetylation than the rest of the gene itself. I'm only working within VWF itself, but we have a couple other lab members working on these ones outside. So that left intron 3 as the first area to look into, on top of the fact that it had that one SNP that was found in multiple different GWASs.
So because it's got these 2 sites, the suggestion is if there's going to be an enhancer region that's going to bind and stimulate VWF expression, this is a great place to look, right?
That's the hope.
Okay. And so you're basically going through and going to use CRISPR to knock down small chunks of that to try and localize where that's located.
Yeah.
And so to my point, can you not just take, how big is that intron 3 region? Can you not,
The full thing is 10 kb.
so can you not chunk some of it and stick it on like a reporter construct and look for regions that,
The the student before me did do deletions of the full 5 kb.
amazing that you remember that.
Yeah, it just suddenly came to me. So
Thanks for the clarification.
sorry.
It's a strobe light or something.
They found that the smaller you went, and they did a 2 kb deletion as well, that the effect was greater.
Okay.
So
Thank you.
Oh, you're gonna say, you were the one unlucky enough to get it. Somebody else got to the other.
what's wrong with it? Drawn 3? It looks great. It's huge.
Yeah. Notifications on. But I think just, I wanna follow up on that because you may not need to take the whole 10 kb. You know, in particular, you know where that snippet is.
Yeah.
And so, functional assays where you just take a small bit and stick it on a reporter, have people trying that with that region. Where you have either allele of that SNP and you see if there's a difference in its.
As far as I'm aware, they haven't done a specific test with just that SNP, but
Yeah, whether that's, I mean, I don't know much about this type of assay, you know.
I should do it. Maybe I should do that. Sounds like a great idea. Thank you, Dr. Brewer.
Well, probably like it, probably because it's pretty easy to do, right? Got the tool, it's silly. Yeah.
Yeah, it's pretty easy to do when you've got a grad student.
I don't like the sound of that.
Did you say you like that or I don't?
I don't like the sound of that.
But just to follow up, that SNP
Yeah,
that was described, it's not in either of your two solids or?
we haven't sequenced intron 3 yet, so
And do we have, and do you have any information on how prevalent that particular SNP is?
I believe it's rare. That's as much as I remember, sorry. I don't think it's a common SNP.
But
Hunter, for your functional validation, what are you using as a positive control?
For
So. Sure, yeah. The CRISPR deletion.
the positive control,
Is there some other part of the region of the BWF gene that if you deleted, you know, you could expect a reduction in expression?
we do have other constructs from the area that have been shown to heavily reduce VDF expression, yes. When we were testing out the alpha genome and trying to validate to see if it worked, we used the aforementioned 40 kb, or no, sorry, it was a small 100 base pair deletion, I believe, just outside BWF that has been shown to have like a 50% reduction on its expression, and alpha genome predicted it was like a 36% reduction in expression. So
That's the 13 base pair.
yeah, yeah.
Thanks.
any other questions? If not, thanks.